Hi everyone, CP welcoming in the new year again with an episode on DNA. This is something pretty much everyone's heard of, and for that reason, there are plenty of misconceptions around it. A great way to introduce this subject is via strawberry DNA extraction. This is a process by which your students will be able to physically see strands of DNA. Strawberries are used because they have a lot of DNA to see. First, they put strawberries in a Ziploc bag and mash them into a fine paste. A rolling pin would work best for least noise and damage to DNA. Next, you add 2 tablespoons of your DNA extraction liquid, which consists of 1 tablespoon of detergent, 1 teaspoon of salt, and 1 cup of water. The detergent bursts open the cell membrane, releasing DNA, and the salt keeps the proteins in the strawberry away from the DNA. Have them continue mashing until it's homogeneous, then strain the mixture with a cheesecloth. You're looking to catch the liquid solution, so a funnel and beaker work great. Take a dropper of the liquid solution, put it into a test tube with an equal amount of alcohol. DNA would normally dissolve, but because DNA is not soluble in alcohol, it will clump together like in oil and water. You should see white strands. These white strands are DNA. When everyone's got the DNA in the tube, ask them how much DNA they think we share with strawberries. They might start at the single digits, but keep going higher until you can tell them it's 60%. Seems like a lot of similarity, doesn't it? A lot of these genes are highly conserved. This means they do things that are essential for life, like transcription or translation of RNA into DNA into proteins. If these proteins were altered in any way, there's a high chance that the organism would simply die. DNA stands for deoxyribonucleic acid. It consists of a double helix connected by base pairs. Base pairs are 5 molecules known as adenine, guanine, cytosine, thymine, and uracil, A, G, C, T, and U. A and T join as well as C and G. One strand will have the pair that matches exactly to the other pair in what's called complementary base pairing. For example, if one side has AAG, A pairs to T, and G pairs to C, the other side will have the sequence TTC. We'll discuss the U later as it's used in RNA or ribonucleic acid. As the name indicates, it's DNA with an oxygen added and serves different functions. Overall, a sequence of base pairs that can be inherited is called a gene. When DNA is ready to be translated to proteins, an enzyme cuts it in half, or 2 strands. Think of it like a zipper, where either end are strands, and zipper is an enzyme. One of these strands is transcribed into an RNA form using the other side as a template. For example, if the AAG strand is read, the other side will be translated into a protein which will be UUC. This is because RNA uses U instead of T, so U pairs stay instead of T. Next, the RNA is translated into sequences of 1 of 20 amino acids in a chain. 3 bases like AUA or GCG are called a codon and equal 1 amino acid. In our example, we can check the amino acid chart to see that UUC gets us phenylalanine. So the bases are translated to amino acid chains, and these make up the proteins that make up pretty much everything in our bodies. This is known as a central dogma of biology. DNA is transcribed into RNA is translated into proteins. A mutation is when a base pair or a sequence of them are placed incorrectly. For example, instead of AUA, your body places an AUG. This means that across from it will be a UAC instead of a UAU. It means different amino acids could be produced. This process can produce alleles. Alleles are different versions of genes. For example, if a gene coded for hair color, there would be a dark-haired allele, a blonde allele, and so on. When you're born, you have 2 alleles of each gene, one from each parent. Which one is expressed in you depends on several factors. Some alleles will be dominant over others, which means if they are present alongside another allele, you will express the dominant one. This state of both alleles being different is known as being heterozygous. Others are recessive, which means they will only be expressed if it is from both alleles from your parents, which is known as being homozygous for a trait. Recessive alleles tend to be less common than dominant alleles, but this depends on the environment. Now for some questions. Why doesn't everything use RNA? Isn't turning it into DNA extra work? Well, yes, but the double-stranded nature of DNA makes it more stable over long periods of time. This is why you can find DNA in the hair and nails of long-dead corpses. Also, due to semi-conservative replication, which refers to how two DNA strands are copied by splitting the double strands and adding complementary base pairs to each to form two new ones, DNA errors can be repaired using the other strand, while if a base pair in RNA is switched out with another, the mechanisms have no way of knowing what the correct one is. Imagine if you knew you'd have to have equal forks and spoons in a table set. If one fork was missing, you'd be able to tell because you'd have one less fork than the spoons. However, if a fork and a spoon were missing, you wouldn't know because nothing indicates they're gone. Why are most mutations bad or neutral? Imagine a very complex computer. I take a ball and toss it as hard as I can at the computer. What's more likely, that I break the computer and stop it from working, I don't affect the computer at all, or I hit the computer in such a way that it functions better? The first two are definitely more likely. To create complex structures like the eye, it's generally thought that each stage of the developing eye has to confer some kind of advantage, or it would be selected out beforehand, but work's still being done on this. What does it mean to damage DNA? Damaged DNA is, by definition, altered in terms of transcription, translation, or coding properties. If you've ever heard the terms free radical or oxidant, these can be created by radiation and UV light like the sun and cause DNA damage by reacting with the backbone of the DNA itself. If this DNA is red, it can produce non-functional or dysfunctional proteins, which can go on to cause conditions like cancer. Antioxidants are considered healthy because they get rid of free radicals. How does the extra oxygen make RNA different from DNA? Oxygen is reactive, and its presence on RNA creates another site for chemical reactions to occur. The fact that it's only a single strand also makes it more vulnerable to enzyme damage. Double-stranded RNA does exist, but it's rare. How can mutations do nothing? If a mutation occurs in a place that isn't coded, then nothing might be expressed differently. Also, you may have seen in the amino acid chart that multiple codons can create the same amino acid. This is so that if the first two bases of the codon are correct, you have a pretty good chance of arriving at the same amino acid even if the last one is wrong. Finally, even if the wrong amino acid is produced, it may be similar enough to its correct form in terms of charge and size that it can fulfill its role anyway. This is known as wobble. We make amino acids, so why do we need essential amino acids from food? Essential amino acids are not produced by the body by definition, and are required for normal functions. However, non-essential amino acids are also helpful to have more of, even if your body can produce them naturally. Now for some misconceptions. Genes don't solely determine traits. You could have the gene for something, but it won't be expressed unless another gene or something in the environment is present as well. This is known as epigenetics and drives the concept of being at risk for certain conditions. For example, you may have a gene that blocks lung cancer, but smoking introduced DNA changes that block that gene, causing lung cancer. Substances that have the ability to induce permanent DNA changes are called mutagens and include radiation and certain chemicals. You generally want to avoid these, but sometimes we can use them for beneficial purposes. Mutation breeding is a strategy used in plant breeding where large amounts of plants are exposed to mutagens and any with beneficial mutations are bred. Most traits are multiple genes interacting instead of just one. This is why it's so hard to pinpoint exact genes for things like hair color, and the example I gave when discussing alleles was greatly simplified. There's a concept called incomplete dominance, where a trait results from two different alleles interacting. For example, if one allele codes for red petals in a flower, and another codes for yellow petals, neither will be fully dominant over the other, and the red and yellow will mix to form orange petals. In a larger sense, many traits could function this way, and removing the function of one gene might alter the expression, but it won't be the only factor. Not all DNA is translated into proteins. Only specific segments of the gene are transcribed into RNA and proteins. The rest is known as non-coding or junk DNA. Segments of non-coding DNA that are removed before translation are called introns, while the parts translated are called exons. Despite the name, these segments of junk DNA play roles in gene expression. RNA that isn't translated into proteins, such as siRNA and miRNA, can perform functions like regulating gene expression as well. Food doesn't damage DNA in most cases, but eating food cooked at excessively high temperatures like burning or frying can do this. Possibly. It's It's related to the consumer incorporating the damaged DNA of the food into themselves, and has been observed in cells and mice, but not in humans yet. Still, good to be aware of. This one is a historical misconception. You might hear that James Watson and Francis Crick discovered the double helix. They did not actually discover the DNA, just the two of them. It was Rosalind Franklin who discovered the double helix, and whether her research was stolen or not, I don't think she gets nearly enough credit for her work. So, once you identify a gene mutation that causes a condition, you can just go on in and fix it, right? Well, yes and no. Genes cannot be fixed, at least in current conventional medicine. The best we can do is to know for the future that a particular mutation causes a condition, and if someone has a mutation, they are at risk. However, innovative therapies such as CRISPR gene therapy may be able to do this with certain conditions in the future, but because multiple genes may be affecting a single condition, it's hard to say if it'll work on everyone. It's like identifying a storm-prone area. You can't change the storm itself yet, but you can build structures accordingly, or even avoid it entirely. On this topic, inserting foreign DNA into an organism and getting them to copy it isn't as easy as, say, injecting yourself with foreign blood. For one thing, the body needs to know how to copy it, or it might just get digested by enzymes. Also, your body might reject it entirely, such as with organ transplants not done properly. For this reason, foreign DNA needs to be inserted via what's known as a vector, or a circular DNA fragment with instructions for copying. To close, you can have students discuss memes. Yes, memes. The definition of a meme is an idea that's transmitted similarly to a gene, in that it self-replicates and mutates. It's an attempt to translate culture into biological terms. For example, someone posts a picture of a dog. Someone else reposts that picture of a dog with a caption. Someone else writes a new caption, edits the dog to look like an anime character, and posts that. Maybe that kind of meme takes off, the anime dog. Someone else might have tried to take a cat in the same pose, but it didn't work, and dies off. The most successful memes replicate themselves just like genes. In fact, memes are sometimes known as the DNA of the soul for this very reason. So next time you share or make a meme, just remember, it's using you to spread itself. And that's all I have for today. Thanks for listening as usual. Sources and transcript will be on my WordPress at helpimteaching.wordpress.com. You can find me on Blue Sky and Twitter. See you next time, and keep teaching.